My name is Drew, and in this video, I'll be briefly going over the founding of our genetic code, specifically the experiments done by Marshall Nierenberg, Heinrich Mattei, and Philip Lader. This is for the prompt 32b. Our journey starts with Nierenberg and Mattei in 1957. They had been formulating a hypothesis that somehow RNA and protein synthesis were linked together. They started their experiments by eliminating other suspects for protein synthesis. They made a solution of tRNA, ribosomes, and amino acids to see if these alone would create new proteins. Did it work? No, it didn't. Interestingly, they knowingly left out mRNA. They were indeed onto something. In this extremely simplified drawing of the experiment, complete with terribly and shamefully drawn stick figures, we can see that they chose a poly-U mRNA and mixed it with E. coli extract. After examining the results, a phenylalanine amino acid chain was found, indicating that the nucleic acids on the mRNA code for a certain amino acid. They repeated this several other times and found that the poly-A RNA created a lysine amino acid chain and poly-C RNA created a proline amino acid chain. These were all very important findings that essentially made the researchers literal celebrities. However, there was still work to be done, given that they didn't know how many nucleotides determined each amino acid. Nierenberg and Philip Lader joined together and conducted another experiment consisting of them washing ribosomes and mRNA with one codon and tRNAs with various amino acids attached through a cellulose filter. Whatever codon chosen, in this case GUU, would attach to its own amino acid. Their experiment concluded that their chosen codon GUU attached to the tRNA containing valine, and thus they were able to make an entire codon table, one that we see in use every day by testing various sequences of codons. Lastly, here are my references, and thanks for watching.